Our next speaker tonight is Shangzheng Huang from the State Key Lab of Brain and Cognitive Science from the Chinese Academy of Sciences.
Okay, thank you very much, everyone. It's a great honor to be here today and share my presentation with you. The topic of my talk is a comparative study of cross-modal and cross-species cortical transcriptomics. As we know, during brain development, complex generative regulatory programs such as chromatin modifications, transcriptional factors, and signaling pathway regulations determine the gene expression profiles of specific cell types. This process follows like the molecular fundamentals for the formation of neural circuits, structure organizations, and brain functions. This molecular fingerprints also preserved into adulthood, maintaining a distinct spatial organization across the cortex. This spatial genes expression pattern has been shown to be correlated with specific functional phenotypes, and importantly with others in disease states. This spatial correspondence can be quantified by computing the spatial correlation between gene expression map and imaging drive phenotypes, which commonly referred as imaging transcriptomics. However, the spatial distribution of genes in the brain can reflect two fundamental properties, stability and flexibility. Stability refers to genes whose spatial expression patterns are highly consistent across individuals, while flexibility refers to those with more variable patterns. Genes with stable spatial organizations often quantified using the differential stability scores or DS scores, they showed higher inter-individual reproducibility. These high DS genes are frequently enriched in cold functional modulars of the brain and are implicated in various cognitive disorders. Moreover, the co-expression patterns among these stable genes are also thought to contribute to the communication across large-scale brain networks. However, so far, almost all imaging transcriptomics studies, including the calculation of the DS scores, have been based on the classical OHBM datasets. This dataset offers relative high spatial sampling resolution and allows gene expression data to be mapped onto any brain atlas via MNI space. For example, using a toolbox like AppGene. As a result, it has been widely used for studying spatial gene expression patterns. Recently, the emerging of single nuclear data sequencing data set covering multiplied brain regions also open up new opportunities for imaging transcriptomics research. However, a key question remains, do the co-function genes in the brain also show consistent spatial expression patterns across these new datasets? To address this question, we first compared inter-regional gene co-expression patterns as well as the similarity of regional gene expression vectors between the two datasets. We found in both co-expression metrics and the regional gene expression profiles show notable cross-modal similarity. Next, we further examine the similarity of spatial gene expression patterns between the OHBM datasets and the single nuclear datasets. For each gene, we computed the cross-modal spatial consistency defined as the Pearson correlations of its spatial expression patterns across the two datasets, and referred to as the CS score. Interestingly, we find a significant correlation between the CS scores and the DS scores, suggesting that spatial stable genes tend to maintain consistent expression profiles across different transcripted datasets. Building on this, we promote a cross-modal data integration framework aimed at augmenting the sample size of the OHBM datasets. This framework includes atlas alignment, stable gene field, and weight combination of expression vectors. After simple augmentations, we trained a regional supervisor deep neural network to identify genes that are highly predictive of cortical populations and to drive regional specific lantern embeddings. We think this lantern embedding captures the value contribution of each gene to regional specific. At the end of training, as a validation step, we extract the gene-wise weights contributing to the network's classification. We find that high-weight genes in both cortical and subcortical regions are significantly enriched in molecules related to core brain functions, including synaptic signaling, ion transport, neural projection, and so on. This indicates that deep neural network effectively learn presentation of region-specific core functional genes in the brain. Furthermore, we want to examine whether the region-specific latent embeddings could effectively capture the brain's transcriptional organization, which is often defined by transcriptional gradients. To this end, we applied a PCA decommutation to the transfer latent embeddings, and we found that the first stringed components explain over 68% of the variance in cortical transcriptional organization. Specifically, PC1 shows a translation from the frontal to visual, and PC2 similarly has a translation from the visual and temporal to frontal lobe. And interestingly, PC3 we find is transition from visual and sensory motor to high-order associated regions. Its spatial distribution aligns with known human macaque brain expansion patterns, suggesting an evolution-related transcriptional gradient. We also find our gradient is similar to recently published articles by Deer et al. Finally, we were also very interested in whether these cortical transcriptional gradients are to some extent conserved across different species. To explore this, we directly apply the DNA drive gene weight and PCA weight to the mouse transcriptic datasets. And interestingly, we find that gradient 1 and gradient 2 derived from the human model exhibit highly consistent spatial patterns in both in human. Yes, and however, gradient range, which is associated with evolutionary expansions in human and macaques, showed clear cross-specific divergence, indicating that this component may reflect specific adaptations rather than conserve transcriptional programs. Finally, to summarize our research, first we find that gene with cross-subject spatial consistency, which means DS scores extend also stability across data sets. Second, in the human cortex, the expression of core brain genes is organized along the string transcriptional gradients, which the first two gradients converge across species. Third, the weight of core brain genes can be learned by a deep neural network model and present as regional specific transcriptional embeddings. Okay, that's all my session, oral section. Thank you. And I will also want to express my gratitude to my innovative team, led by a young but charismatic Professor An Li. And I also want to thank the Institute of Biophysics Chinese Academy of Science. Thank you. Thank you for your attention. Any question?
Yep, so thank you for a very nice presentation as well. The cross-species differences that you found, did you also look at differences between macaque and mice, or do you always use the human as the comparison?
Okay, thank you for your constructive questions. Indeed, we have not to let research.
Okay, yes. So you looked at human versus macaque and human versus mouse?
Yes, human versus mouse. macaque is just compare the PC strand map with the cortical expression maps.
Okay.
Yes.
But you didn't see what the differences were between the cortical expansion map of macaque and mice?
Oh, no.
No. Okay. So
okay.
that
Very okay.
I was just wondering if the degree of difference would be the same as with humans.
I think the macaque map may be very similarity with human than mouse. Yes.
A greater one. Well, that's no more. Do you have a question?
Yes, yeah, thank you very much.
Thank you.
I was wondering how you accounted for some of the cell population and the proportion of cell populations that you may have in the different samples. I think, as you mentioned, the Allen brain samples very precisely, while the single cell data can consist of many different cell populations. So did you account for that in your comparisons?
Okay, the HBA datasets and the single nucleus datasets did have value difference. So when integrated these two datasets, we consider the consistent genes patterns in the brain. We filter this gene and we do a italize alignment, and by this we weighted the two datasets of the gene expression. Okay.
Thank you.
Okay. Thank you.
Thank you very much.
Thank you very much.